And thank you a lot for this opportunity to present our SDG solution. And our pharmaceutical company is based in Kyiv, the capital of our country. And during the Young SDG Innovation Program, we tried to find the answer for a few challenges, but finally we focused on the evident problem. Most of us watched how the world map changed the color and coronavirus became the main topic until today. But our team want to transform this pain into gain, and first of all, we looked back how it was. We start research information about respiratory virus infections we had before COVID pandemic, and found that main players on this stage were flu A and flu B, well known as influenza, and the respiratory essential virus, rhinovirus, and enteroviruses. And this data is confirmed by statistic organization from Europe Union and United States of America. And it was insightful for us that there are no information about RSV in Ukraine at all, and we understand that it's good time to solve this problem. The severity of RSV infection is so critical for newborns and the elder people, and according to the data from US Center of Disease Control, the average quantity of acute RSV cases is 177,000 per season, and the mortality visualized easily as a football stadium full of people. For every 100,000 hospitalization, 9,000 die. As I said before, newborns is in a high risk of RSV2, and this summer RSV outbreak in Dutch hospital proves it. And as you see as a slide, the monitoring of respiratory essential virus in United States perform continuously, and they perform the trends, but in our country there are no instruments like this. And our challenge is to improve the effectiveness of respiratory viral infection diagnostic in our country. And the size of opportunity is above 5.4 million tests, and that will help Ukrainians to have right acute respiratory infection diagnosis in the right time. It helps 12,000 of newborns don't have a severe RSV illness as a pneumonia. And our challenge solution have direct impact on SDGs as a good health, innovations, and infrastructure, and partnership for the goals. And our main stakeholders in this challenge was and still are the citizens of Ukraine, the Ministry of Health, the State Institution of Public Health, Farmak as the main business stakeholder, and test laboratories around the country. And about our solution in detail described my colleague Ihor, the subject matter expert in our solution.
Thank you. Okay, our solution is a diagnostic PCR testing kit for detection of several respiratory viruses. And now many people have some experience with PCR testing, but usually most of them participated only on the first stage, first step, that is sample collection. Our product is responsible for the last step, where the detection happens. And the best PCR kit for laboratories is the one that detects more targets in one reaction. That is why we are working on the PCR test kit that can identify the presence of any viruses from this list. Diagnostic kits are absolutely new type of product for our company, but on our way to the best solutions, we have already gained some results. We were successful in developing and manufacturing PCR kits that can detect SARS-CoV-2. Also, now we have proof of concept prototype that can detect influenza viruses and coronaviruses in clinical samples simultaneously. And the last stage, we are now developing the test kit that can identify RSV as a new target together with above mentioned viruses. So step by step, we are going to our main goal. Our work is not finished, but we believe that it will be a good contribution to the public health. And thank you very much for your attention. If you have any questions, we will be happy to answer them.
All right, awesome. We have 2 minutes and quite a few seconds to spare. So and we'll just have quick questions from our judges and any comments as well.
I do have a clarifying question on the solution. I thought it was a great presentation. I just want to understand a little bit more. What is the difference between what this solution provides and what is available today in terms of maybe speed or cost or some other variable?
There are a few things. First of all, in Ukraine, for example, during pandemic, we have some not so many variants of tests available on the market. First one. Secondly, our solution is about many targets in one reaction, and it needs some know-how to develop such kit that can detect it in one reaction. There are some available tests in the world that can detect different viruses, but they usually are separated in different reactions. And also the other part that is important for Ukrainian market is the price. So we are focusing on these main points for our market. That is why we investigated our market. We interviewed different laboratories, opinion leaders, what they need, how they need, and so on.
And the kit itself is on site at the medical facility? Is it sent out to be for the test, or is it on site?
This project is separated in different steps. Some steps are resulted in product that is now available on market. It's about SARS-CoV-2, but we are developing the more complex product that can detect different viruses simultaneously, and these stages are still going on.
Okay, thank you.
All right.
Victor, is there time for one question? Yeah.
Yes, please.
I was curious. You mentioned in your presentation that this is a new area for Farmak, and I'm just curious to know, like, what other things have to be put in place. Are there new relationships you had to build? Like, if you're moving into a completely new area, are you set up, or is it going to be easy for you to roll it out?
Well, this project for Farmak is not only a new product, but a new way of developing, because usually the products that are more traditional for our company, they are developed during many years. They are absolutely different technologies, and so different experience you need. And in this situation, we have a short time. Our team is somehow separated from the common processes in company, and we are also like a project inside the company about how to make things differently.
Thank you.
All right, awesome. Mary?
Thank you, great presentation. I think my questions were answered through Joseph and Helen, so no worries at all. Good work.
All right, brilliant. And we're a bit ahead of schedule, so I think we can take one question from the audience. Anna is asking, please give more details about tests. Thank you in advance.
What kind of details? Because the discussion can be another presentation, another lecture. So,
of course.
In general, it's a test for laboratories that can perform one PCR reaction from one sample, from one person, and detect what kind of virus causes the respiratory problem in this patient. So to distinguish which is the problem, we now know about coronavirus a lot, but there are still other issues that we need to consider.
I had a similar. I was curious as well about, is it like a blood test? Is it like a nasal swab? And is it following a similar procedure as other tests that already exist in the system? So there aren't a lot of new processes or protocols, and it's fairly stable, and it can be done in an environment that already exists. I was curious.
In this, from this point of view, it's traditional test. So it's swab because it's respiratory viruses. So the best way to take the sample is to take it from swab. So it's the same as for coronavirus.
All right. I believe all questions have been answered. Thank you very much. Thank you very much. Great
Thank you.
presentation. And we look forward to seeing you guys save the world.
Thank you.